So today I'm going to be talking to you about my PhD, which focuses on the genetic basis of host specificity in Pseudomonas syringae, using the strains which cause bacterial canker of cherry and Prunus to explore this. So I've included these two symptom pictures on my title slide. Here's a picture I took up at the Rocks Orchard in February, and we have a particularly bad case of canker with the large black necrosis and extensive gummosis beneath the tree. And here's another picture which was taken at EMR in 1980 of plum dieback due to canker, where the cankers occurred further up the branch and caused dieback. And EMR was the place where a lot of the pioneering work on canker was originally done. So I'll just run you through the contents. I'll first introduce Pseudomonas syringae and the topic of host specificity, then go through some aims, some preliminary results, and the future work. So Pseudomonas syringae is a rod-shaped gram-negative bacterium, as you can see from the electron micrograph. And it has a very broad host range and infects over 180 plant species, including many of the important crops seen below. It's not only a plant pathogen, however, it can be found in environmental reservoirs such as water, which it uses for long-distance dispersal around the world. Pseudomonas syringae can be divided into groups of strains which cause disease on particular plants called pathogenic. For example, the pathogen which causes disease on cherry is called Pathogen morspinorum. And these pathogens can be further divided into races, which are strains which show differential virulence on particular cultivars. And Pseudomonas syringae has global economic importance. There's two diseases which in recent decades have been causing problems. The first being kiwi fruit canker, which has devastated the industry in New Zealand, and the second being horse chestnut canker, which has been a real problem in northwest Europe, where hundreds of thousands of trees have been infected. And Pseudomonas syringae is a model pathogen in the study of plant-microbe interactions, and it was voted the top bacterial plant pathogen in 2012. Okay, so the disease I'm focusing on is bacterial canker of Prunus, which is caused by two pathogens, Pathogen morspinorum and Pathogen syringae. And there are two races of morspinorum due to specificity towards particular cherry cultivars. And here's a simplified diagram of the life cycle. So first, in spring and summer, the pathogen colonizes leaves epiphytically, and then particularly virulent isolates will infect the leaf via the stomata, causing a leaf spot. And then when the temperature cools in autumn and the leaves fall off, you get infection via leaf scars or natural or man-made wounds. The bacteria then overwinter and cause canker, where you see a large black necrotic region with usually seeing gummosis around the region. And then when the season progresses, the bacteria come to the surface and are dispersed via rainwash. So Pseudomonas syringae contains both pathogenic and non-pathogenic isolates. And the key difference to this is the presence of a type 3 secretion system. So this is a macromolecular complex which spans both the inner and outer membranes of the bacterial cell and has a pilus which extends to the host cell which is used to inject bacterial effector proteins. And this electron micrograph shows the pilus, immuno gold staining, and the secretion of an effector at the end. So bacterial effectors are key to Pseudomonas syringae's pathogenicity, and effectors are proteins that alter host cell functioning. They mainly act to suppress plant defences and to aid the bacterial's dissemination throughout the plant. So this diagram just shows some of the host cell targets of the effectors, and we have a virulent isolate here injecting effectors, and these effectors target multiple signalling pathways within the cell, and a non-pathogenic bacterium which hasn't been able to introduce these effectors is killed by antimicrobial compounds. So to be able to respond to pathogens such as Pseudomonas syringae, plants have a highly complex and robust immune system. This consists of preformed defences, such as the hydrophobic cuticle, which surrounds the aerial surface of the plant and prevents the initial attachment of many pathogens, and induced defences. These induced defences are divided into two layers and can be triggered by two different types of molecule. We have PTI, which is PAMP-triggered immunity, which is triggered by the recognition of conserved pathogen molecules. For example, the bacterial flagellin is a conserved molecule important in the movement of bacteria, so is a PAMP, which triggers this plant immune response. And then we have effector-triggered immunity, which is important in the recognition of the effectors within the plant host cell. And this picture just shows two tomato plants infected with Pseudomonas syringae. We have a resistant plant which has a robust immune response and has been able to prevent disease, and a susceptible plant where the bacteria has successfully suppressed the host immune responses and caused disease. So in 2006, Jones and Dangle devised a model to be able to explain the interaction between effectors and the plant immune system. So when a bacteria or most pathogens invade, they produce PAMPs, which are the conserved molecules, and the plant can detect these via pathogen recognition receptors, which here we have PAMPs, the PAMP flagellin activating the PRRs, and this leads to PAMP-triggered immunity, or PTI. A successful pathogen will be able to suppress this PTI, and here we have two effectors there, inhibiting the functioning of PRRs within the host cell to cause effector-triggered susceptibility. And then moving on, in resistant plants, they will produce resistance proteins, or R proteins, which are able to detect the presence of effectors and trigger effector-triggered immunity. And this is typically associated with the hypersensitive response, which is a programmed cell death mechanism to prevent the spread of the pathogen throughout the plant. And then if we carry on with the model, the plant will, oh no, I forgot where I am. The pathogen will then be able to evade this effector-triggered immunity. It will either do this by deleting its effectors that have been detected or modifying them. It will suppress this PTO-mediated immunity, or it will produce effectors which target other areas in the pathway to lead to effector-triggered susceptibility. And then finally, a plant will then respond to be resistant by producing R proteins, which are able to detect the presence of these new effectors. So this co-evolution between pathogen and host has led to pathogens with very large effector repertoires within the host, which act within the host cell, and they are functionally redundant, which means that many of them perform the same functions to produce the same outcome. So going back to this diagram, we have various host cell targets targeted by multiple effectors. So various effectors target the PRRs, several target the MAP kinase cascade. And this means that each strain has its own unique repertoire of effectors, as it has fine-tuned its effector repertoire to maximise pathogenicity to a particular host. And therefore it's hypothesised that particular combinations of effectors may be important in both pathogenicity and host specificity. So my project is looking at how host specificity of a pathogen is controlled, and how a change in host specificity determinants may allow a pathogen to evolve to be able to infect a previously resistant host, a phenomenon called host jumps. So So plants are generally resistant to most pathogens, and what this diagram shows is that a pathogen's host range is excluded to only those that it has encountered, those hosts that it's encountered, and those that are compatible with it. And pathogens can be divided into generalists, which have a very broad host range, and specialists, which have a narrow host range. Pseudomonas syringae, with its broad host range, you might think it's a generalist pathogen. However, individual strains can only infect one or a few plant species, so it's actually a specialist. And host specificity is controlled at multiple levels. It can be controlled during the initial attachment, during the colonisation of the plant, and also during the complex interactions between the effectors and the plant immune system. So it's hypothesised that a change in these host specificity determinants will lead to the evolution of host range as a pathogen is able to adapt to a previously resistant host. And you would think that these host jump events would be rare, because a pathogen, as it adapts to its host, it's fine-tuning its effector repertoire and therefore becoming less and less able to infect another plant species. However, in Pseudomonas syringae, the host jump events are rather common. So why am I studying host specificity? It's become increasingly apparent in the recent decades that the major threat to our agriculture and forestry is emerging diseases, and the BBC presented this article showing that the major threat was unknown pests. And with global warming, the movement of crop pests and pathogens towards the poles and increasing global trade of plant material is increasingly exposing pathogens to new environments and new hosts, which may drive the emergence of new diseases and evolution of pathogen host range. By studying host specificity and learning more about it, we may be able to find management strategies which will prevent this rapid evolution, as well as potentially be able to predict new hosts for a pathogen. So, in Pseudomonas syringae, which I'm focusing on, I've created two phylogenetic trees, which are a little bit blurry. And so this is the pathogen, and this is the corresponding host, and they are labelled by plant order. So if we expect host specificity to be vertically inherited and phylogenetically related strains will infect phylogenetically related hosts, we would see congruence between these two trees. And we see this here with 3 related bean pathogens infecting similarly related bean hosts. However, if we look at the rest of the Pseudomonas syringae phylogeny, we find very little correlation between the relatedness of the strains and which host they infect. With cherry, we find that 3 highly divergent groups of strains infect this plant species, and therefore these strains have convergently evolved to be able to infect this host, so must share host specificity determinants to be able to cause disease. So the aim of my project is to explore host specificity at several levels. To do this, I will be sequencing the genomes of several strains and using comparative genomics to try and find differences in these host specificity determinants. The scales I'm looking at the pathovar level, which is between the different cherry pathovars seen here, and these have all convergently evolved to be able to infect, so I'll be looking for similarities in host specificity determinants. At the race level, so this is between the 2 more spinorum races which show differential virulence on particular host cultivars. However, these 2 groups of strains, although they've been classified as the same pathovar, it's clear from this phylogeny that they are very distinct organisms and phylogenetically unrelated. And finally, at the host level, so here we have a group of cherry strains, and we also have a strain which has been isolated from the perennial shrub Aquilegia, and Their relatedness suggests that there may have been a host jump between Acknowledgements and cherry, and I'll be sequencing these strains to see if I can find effectors that have been involved in the host jump. So my first two aims are to characterise my bacterial strains and to determine the relationships between them, to assess the pathogenicity of my different strains, to try and find differences in virulence, and also to find if race structures do exist with particular strains causing disease more severely on particular cultivars, and to sequence the genomes and begin the comparative analysis. So I started by characterising all the strains that I received, and I did this by using a PCR method called REP-PCR, which it uses primers based on repetitive extragenic repeats within the genome. And this produces characteristic banding patterns on the gel, which enables you to identify different races and pathogenic accurately. So here we have Morspinorum race 2, and we're able to say that these strains are Morspinorum race 2 due to their banding pattern. And then to assess the relationships between my different strains, I used multi-locus sequence typing based on 4 housekeeping genes within the bacteria. I used PCR and then sequenced the products and then conducted an analysis on Genius. And this is one of the trees that you saw earlier, which allowed me to assess the different relationships between my groups of strains and be able to find the number of strains I would sequence, as I would want representatives from each of these 4 groups. I then wanted to assess the pathogenicity of my strains. I first focused on Acknowledgements, and I wanted to produce an assay which would reliably produce these lesions on the leaves. However, it required quite a lot of optimization. I needed to optimize parameters such as light intensity, humidity, temperature to be able to get my pathogen to cause disease. And eventually I got the Acknowledgement pathogen to cause some disease lesions. And to validate whether or not this was disease, I did a population count experiment where you measure the population growth of the bacteria within the leaf, and the Acknowledgement pathogen is in green and has increased to high levels, and then 2 non-host pathogens, the cherry pathogen and the tomato pathogen, did not reach these levels. And here we have the inoculated sites. You have the control, the cherry pathogen, which is the one related to Acknowledgements, which doesn't cause disease, the tomato pathogen, and then my Acknowledgement pathogen causing a disease lesion. And then to assess pathogenicity on cherry and plum, it's more difficult because canker only occurs in winter and spring. I've used an in vitro cut shoot assay where you take second-year shoots from a tree and inoculate them with the bacteria and then measure the length of necrosis. I aim to use detached leaf and fruitlet assays in the summer to assess pathogenicity. And finally, I have grafted 360 cherry and plum trees, and we'll be doing whole tree experiments next year using wound and leaf scar inoculations. And this will allow comparisons between isolates in terms of their virulence and host cultivars in terms of their susceptibility to be made. And here are some examples from the in vitro cut shoot assay, whereby we have the green healthy controls and the necrotic lesions on the infected. And I measured length of necrosis and ranked the percentage area, and a more accurate measurement will be produced on image J. And finally, I will be sequencing the genomes of 16 isolates using aluminum MySeq paired end 300 base pair reads. For anyone that doesn't know very much about genome sequencing, you have your genome and you fragment it to 600 base pair fragments. You then ligate adapters onto the end, and then you put it into the aluminum machine, and you get a sequencing read of 300 one way, 300 the other way, and you get 600 base pair reads which you assemble into a genome. So currently I am optimising a library preparation method, which is these steps here, based on an inexpensive protocol produced by Wilkening et al. In 2013. So for my future work, I will continue the pathogenicity tests, sequence the genomes of my isolates, assemble them and begin annotating them. I will then use comparative genomics between my isolates to try and find effectors involved in host specificity. And then when I have candidate effectors, I will be able to try and manipulate host range within the lab by transferring these candidate effectors into different strains to try and perform a host jump. And these are my acknowledgements by 2 supervisors and organising committee. And thank you.